Hi, my name is Brittany Carpenter, and this is my description of a cell. So I'm going to draw it as a circular shape. It can be square, it can come in all sorts of different shapes and sizes. That's just an easy way to draw it. Um, this outline is the plasma membrane. The plasma membrane is made of a phospholipid bilayer. So if you were to zoom in on this and make it bigger, you would see that you had the phosphate heads with the fatty tails, and the heads are to the extracellular fluid and the intracellular fluid. All the tails point into each other. Also in the cell, you're going to have a nucleus. The nucleus is sort of enclosed by this, again, a double layer membrane that's called the nuclear envelope. And then inside the nucleus, you'll have the nucleolus. The nucleolus is where the ribosomes are held. The nucleus is where DNA is stored, so that's all our genes and everything like that. The nuclear envelope has pores in it that are continuous with the rough ER, which that's a horrible drawing. The rough ER, it's called the rough ER because it has ribosomes all surrounding the outside of it. So it makes it look bumpy when you look at it. The rough ER is cisternae, which is just another word for saying tubes. So in these tubes, the proteins come from the nucleus and travel through these and are sort of being made. And then from the rough ER connects to the smooth ER. And the smooth ER is called smooth because it doesn't have the ribosomes on the outside. And so again, it's a cisternae where the proteins continue to go through their production. Another organelle that we have are the Golgi complex. So this looks similar to a stack of pancakes, like that, and then you'll also see these vesicles sort of budding off and nearby. So this is the Golgi apparatus, or Golgi complex, and these are Golgi vesicles. The Golgi complex sort of packages and does the last final touches on the proteins, and then they are transferred to the vesicles, which the vesicles can either take the proteins outside of the cell, exocytosis, or they will transfer them to somewhere else inside the cell, which is endocytosis. So we've got lysosomes. Lysosomes are another type of vesicle, but these ones go around and sort of clean up the cell. Any molecules that aren't being used, that are dysfunctional, the lysosomes break those apart. Peroxisomes, same kind of thing. Breaks down molecules, but it uses hydrogen peroxide to do so, whereas the lysosomes use proteins. And we also have mitochondria. Mitochondria are usually pretty easy to find because they'll be sort of oval in shape and they'll have sort of like lines going through it. The lines are actually part of the membrane. The mitochondria is where ATP, which is the energy that the cell uses, that's where energy is produced. And then we also have centrioles. Centrioles are important for cell division. So centrioles, you'll always find two of them in the cell, and they'll be found perpendicular to each other. So they'll be in the same area, semi, like that. And that just about covers everything. Thank you.